Well, I guess my lecture might be boring for the bioinformatics, but the topics I'm going to cover are very important for the data they're going to be evaluating, because if your RNA or your sample is garbage, then the data you're going to get out is garbage. So it's very important to have in mind what things can you do with the samples that you might have available, clinical samples or whatever. So we've all mentioned already, the previous speaker, the central dogma of molecular biology, DNA, is used as a template for RNA synthesis. Then some of the RNA molecules produced are used as a template for protein synthesis. And the number of products from the number of genes that we initially had estimated, this number keeps changing and will continue to change as we can evaluate further the sequences in the Human Genome Project and new data coming out from the resequencing efforts. It's estimated that if there's 3 different mRNAs, which is actually a very low estimate, are produced from each gene, then you will have like 100,000 RNAs and possibly about 1 million different proteins. But these numbers are all very rough estimates because that depends on the cell type and all that. So when we're using microarrays, or even the new methods that are coming out and already established, and the new ones that are going to be made, we're studying mostly what is in the cell. 80% of the RNA sample that you're going to obtain is ribosomal RNA. About 10 to 20% is tRNAs and 5S ribosomal RNAs, and only from 1 to 5% represents the mRNA, which codes for protein. And on top of that, there's other, apart from the classical RNAs, mRNA, ribosomal RNAs, and tRNAs, there's a whole bunch of non-coding regulatory RNAs, like the siRNAs, the microRNAs, long non-coding RNAs, and other RNAs that have been discovered in the past few years that impact the regulation of gene expression. So there are microarrays that evaluate these type of molecules, and the sample preparation is very important, because if you don't use the right method to isolate your RNA sample, you're not going to be able to evaluate microRNAs, and I'll let you know in a few seconds, how that goes about. So the types of expression arrays that I'm going to concentrate in this talk are the Affymetrix platform, which we have at the Medical Sciences campus. It's very sad that, the expectation is that those platforms will be obsolete or are already obsolete, but we have them. That's the important thing. If you want to use them, we have them. And most of the studies that have been done with this platform have been the arrays that use the 3' IVT design, which mainly samples RNAs based on probes that target the 3' end of the RNA. Apart from that, there's the human exon array and the gene arrays. This table comes from Terry Spieth's 2007 paper, where he compared these 3 platforms in terms of their variability, how reproducible they were, if you compare the same sample in the different platforms. It's a very, very nice paper. I hope you guys have looked at it. So depending on the platform, you have different number of probes. This design uses a perfect match. Each 3' end of the mRNAs are sampled using perfect match and mismatch probes. That type of design is not used in all the sequences, only in a set of them in the exon arrays and the gene arrays. The human gene arrays have a subset of probes from the human exon arrays. And they don't give you as much information on alternate transcripts. I'm going to show you some of that now. So what type of transcripts can you evaluate using the 3 different platforms, main platforms for the Affymetrix arrays, expression arrays? The classical 3' assay, the 3' IVT assay, only samples the 3' end and the presumed standard transcripts. But if you have transcripts which have an undefined 3' end, they're not going to have probes representing that gene because they're going to be missing. Messages that are not polyadenylated are not going to be able to be sampled because the assay for the 3' IVT array starts with a primer that recognizes the 3' end. So if you don't have a poly-A tail, you won't have a place where the primer will bind and then elongate. Truncated transcripts won't be detected if the truncation occurs in the 3' end. Samples that are degraded can be degraded either from the 5' or the 3' end. Those that have degradation in the 3' end will not be covered or not be detected in the 3' IVT arrays. So alternate splicing events. The 3' IVT arrays do have some probes for alternate transcripts, but not all of them are covered. If you have a whole transcript assay where all the exons are represented, you will cover all alternate splicing events. Alternate 5' start site may be detected also with the whole transcript arrays, but that depends on the annotation, if it's correct or not in terms of the genes. If all the alternate 5' ends have been already described, then you will have probes for those 5' ends in the array. But if they are new, like the ones that are coming out with all these whole genome sequencing projects have uncovered, like I'll mention at the end, you won't have those probes in the classical whole transcript assays. So let's talk about some practical issues. How much material do I need to carry out a microarray analysis? This is for the biochemists and cell biologists here, those who are actually isolating samples. So for the 3' IVT expression arrays, the recommended amount of template RNA is 100 nanograms, minimum 50, maximum 500. For the microRNA arrays, it's about the same range, from 0.1 to 1 microgram of total RNA. These assays can use total RNA with very little processing before starting the whole the reactions for labeling and amplification of the RNA. Those arrays that use the whole transcript approach have two types of labeling protocols. These two types of labeling protocols are for the exon arrays. The exon arrays, since they're not based on poly-A priming, oligo-DT priming of the synthesis of the reverse strand from the RNA, they require, the whole transcript. They're not using that approach. They're using random primers to prime the synthesis of the complementary DNA of the RNA. So that will cover every RNA that is in the sample. So if you start for the exon arrays with a large amount of RNA, this is considered large for many clinical samples, you have to do a step where you remove most of the ribosomal RNA. That's the ribo reduction step. And it's a very critical and sometimes not very reliable method. The company that makes the kit for removing the ribosomal RNA is Invitrogen, which those people here who have worked with kits from that company know that they're not very reliable. Sometimes they work, sometimes they don't, and you can waste a lot of samples trying to remove ribosomal RNA. They do have an alternate protocol that uses 100 nanograms of RNA as starting material. They don't require there to remove the ribosomal RNA, but you can lose a lot of information that is covered in the exon arrays if you use the 100 nanograms total RNA labeling protocol. The gene arrays, which contain a subset of the exon arrays, do use the 100 nanograms total RNA labeling protocol. Since it's a smaller amount of total RNA you're using, even if you use a random primer-based method to amplify your RNA for preparation, for labeling, you will still be able to detect a lot of information from that array. So in terms of the sample types that you might have, what kind of samples you might have? You might have a blood sample, which is a non-invasive, relatively non-invasive method to isolate a sample from your patient for studies. You can have a tissue sample, which could come from a biopsy from a patient that goes into surgery. The problem with tissue samples is if it's an invasive method, it's very hard to get good controls to compare, normal tissue versus the tumor tissue, for example, in a cancer study. So usually what is done is when they do the surgery, they look for other parts of the organ that is being operated on that looks apparently normal. They have to do pathology to decide then if that's an adequate normal tissue in order to do the comparison. You can also use paraffin-embedded, formalin-fixed samples for isolation of RNA. That's been done. It can give you samples adequate for microarray analysis, but of course, over time, those samples, you would expect them to be degraded and to have some chemical alterations due to the fixation processes. In terms of these type, and also you could have cells. These studies are usually done when you culture cells from a tumor and then use those for your microarray analysis. The problem with using blood samples is that you have to try to stabilize that sample as soon as you draw it, because there's many changes that can occur rapidly in a blood sample that will alter your results. Same thing may happen also with tissue samples. So basically, you need to add something like RNA protect, RNA later, things like that to your sample in order to protect them from degradation. But what happens then, that, for example, RNA later will cause your samples to be harder to disrupt in your processing method. So we'll give you a lower yield of sample due to your stabilization protocol. In terms of RNA isolation methods, there are basically two types. Either you use, or actually three types, but this one's a subset of this one. You can use classical techniques for RNA isolation, which involve reagents like the trisol reagent, which you can isolate RNA by undergoing phase separation. In that type of sample, when you use trisol as a method for RNA isolation, you can isolate DNA, RNA, and protein from the same sample. The yield is not the best for probably if you do RNA isolation, your DNA and protein yield will probably be much lower than you would expect if you did it only for protein or only for DNA. But you can still get a small amount of the three types of substances that usually one wants to study. It's a cheap method. Actually, it's a reagent that is not costly. It does take time. It involves many steps. And at the end, you finish up with your nucleic acid in a pellet. That, if you're not careful, you might lose, which is it's actually, when you work with nucleic acid pellets, it's a problem, especially if it's a person that's not very experienced. And this method is not adequate to study short RNAs because they don't precipitate very well. So in order to get them, you have to do some adjustments. The spin column methods, which are widely used, commercial companies like QIAGEN, Ambion, etc., they all have different spin column methods. They are faster and easier. The purity of the sample is very high. You have to take in mind that depending on the assay you want to do, you might need to have a specialized kit used for isolation. For example, for microRNAs, you would not use the regular RNA-easy kits from QIAGEN, you will use one that's specific for microRNA, so you can ensure you can have that sample that you want to analyze. If the person or the person who is doing the isolation is not well trained, even if you use a column method, you might not be able to get your sample because some people don't know how to, if they overload the column, they will lose the sample. So you need to ascribe to the instructions very carefully, and the cost is much higher, of course, than the trisol method. The automated methods usually use the same technologies as the spin column methods, but in an automated fashion, so that allows you to handle many samples quickly, and they're usually pretty good. So the good thing about the automated methods is that you minimize the variation on how the person handles the sample, which in the spin column method and in also the trisol method, depending on the person, you will have variability on the results and the quality of the sample. The automated methods, well, at least they're all brought at the same, treated in the same manner prior to putting them in the machine, at least whatever comes out is going to be very similar in quality. They do require specialized kits for the machines, and the cost is high. So this is the slide that I wanted to show you in terms of the size of the sample. If you use reagents like the RNA-easy kits from QIAGEN to isolate your RNA, the RNA that you're going to get is higher than 200 nucleotides in length. So all your 5S ribosomal RNA, which usually you don't want to analyze, the tRNAs, which are about 70 nucleotides long, the microRNAs, which are 20 to 30 nucleotides long, you won't be able to isolate them using these methods. So you need to have a specialized kit for microRNAs. In terms of the starting material, this is a table that the QIAGEN company provides, but it gives you a good idea of what the spin column methods in terms of the yield of the sample. If you start with 5 times 10 to the 5 cells, this is a human cell line, you get about 7.5 micrograms. So recently, I've been involved in a grant preparation, and we were looking to see whether, for example, an oral rinse sample would be adequate for carrying out microarray analysis. Well, an oral rinse sample in a paper that I looked at says that you only get about 11,000 cells from an oral rinse sample. That won't be adequate to carry out RNA analysis, even if you use the 100 nanogram or the 50 nanogram method. So you need to think about those things when you decide what sampling method you're going to be using. Depends on the analysis you want to carry out. Okay? In terms of tissues, if, for example, this is something I also was involved in recently, they asked me, okay, how much tissue you need in order to do a microarray analysis? So 5 milligrams from mouse and rat, for human, you would expect it to be about the same. 5 milligrams would give you from 10 to 4 micrograms of RNA. That's in theory. That is, if that tissue, depending on the tissue, you will be able to break it up easier and liberate the RNA from that tissue. So that's why you see the variability. And also, if the tissue has been frozen, stored away in a way that is not really adequate, your yield will also be lower. So these numbers are for probably, freshly isolated tissue. This is the maximum amount that you might be able to get. So you always take into account even 5 milligrams of tissue will, depending on the quality of the sample, may not yield an amount of RNA sufficient for your further analysis. The other thing that I wanted to point out is, okay, maybe I have enough RNA to carry out a microarray analysis. But what happens afterwards? You need to validate the genes that you saw differentially regulated in the experiments you did. So if you only have enough for a microarray, you will not have RNA sample from the original samples used in the study to validate the differential expression that you determine using microarray or whatever method of analysis. So the minimum amount should not be the amount that you need for the microarray, because you need to validate your data. So for example, for real-time PCR, you need to use a minimum of 10 nanograms of RNA per well of a real-time PCR assay. Usually do that in triplicate. You need to validate housekeeping genes plus your test genes. So that really requires like another microgram of RNA just to do the real-time validation. So then let's say, okay, you isolated your RNA. Now what are you going to do with it? You're supposed to store it. The best way to store a sample for RNA analysis is to store it already as purified RNA. Storing samples as frozen samples, for example, at -80, there's many tissues that are the RNA within is so unstable because there's a high level of RNases, like liver, for example, that if you store the tissue, you will not be able to isolate good quality RNA afterwards. So the best thing is to isolate the RNA as soon as you can and store it at -80. If the RNA is pure, it should be stable for about 8 months. This would be a minimum amount. There are samples that last much longer than 18 months. If you store them at -1, like a liquid nitrogen level, you might extend that period much further. The critical thing is to avoid freezing and thawing your sample. Try to, when you isolate it, prepare aliquots after you quantify it, and then just take out a small sample at a time for analysis, rather than the whole sample and then freeze and thaw. That will cause degradation of RNA. And if you have low concentration of samples, you might need to use carriers in order to concentrate them further for the steps needed for the microarray analysis or real-time PCR. So in terms of, okay, now you have your RNA, you stored it. Before you store it, usually you take an oligo out for analysis to see how much RNA you have. First of all, my quantity, the purity, and the integrity. Excuse me. So in terms of the purity, you want to know, usually you use the absorbance measurements. You use absorbance of ultraviolet light at 260, 280, and 230. For integrity, you need to analyze the sizing of the RNA in your sample. And there's basically two main methods, capillary electrophoresis and agarose electrophoresis. And there's a third method, which is the 3' to 5' ratio, which is something you can only do using either quantitative real-time PCR or microarray analysis. So in terms of the quantity, most of the time people use spectrophotometry. You can also use, if you have a very small amount of RNA sample, fluorescent dye methods that allow you to detect up to 1 nanogram per ml in a sample. The most widely used instrument for quantitation is the nanodrop instrument, which you can use only 1 microliter of the sample for quantitation. The detection limit of the nanodrop is about 2 nanograms per microliter, so it's much less sensitive than the fluorescent dye methods. The spectrophotometrical methods are sensitive to contaminants in the sample, which might absorb in the same UV light range as the nucleic acid. And it will not discriminate between RNA and DNA because they basically have the same curve of UV absorbance. In terms of comparison of the two, I would say the fluorescent dye methods are more time-consuming, involve more handling because you have to mix your sample with the dye and then read and then do a standard curve and all that stuff. So it takes more time, it's more expensive, more time-consuming, but it has a higher detection limit. So when you do the nanodrop assay, not only it will give you a value, but if it's connected to a computer, you will also provide a UV spectra, which is very important. You need to look at that UV spectra because this is the shape of what you would expect. So normally, in this range of UV, from 220 to 240, 240 to 260, you have absorbance of substances that are used in the purification of RNA, such as phenol, EDTA, thiocyanate. They all absorb in this range. So if you see your typical curve, in addition, something else on this, like a shoulder on this side of the spectra, you have to suspect contamination with phenol or other reagents. This is where you would expect your RNA and DNA to absorb. Phenol absorbs, which is present in trisol reagent, will also absorb in this range. And tryptophan, which would be an indication of protein contamination of your sample, would absorb further to the right in the spectrum. Not only do you have to look at the shape of the curve, you also have to look at the intensity of the range where the RNA and the DNA samples are absorbing. A sample that is of good quality will show a spectrum of this type. Samples that are partially degraded will have a decrease in the 260 region, and if they're of very bad quality, that shoulder will practically disappear. So you have to look at the spectrum. Another thing that you have to do with the UV data is look at the ratio between the 260 and the 280 wavelengths absorbances. It should be around 1.8 to 2.0. If it's different from this number, usually it's lower when it's RNA that is of low quality. It's lower than 1.8. It will indicate protein and phenol contamination. Remember that even this value doesn't discriminate between RNA and DNA. So even if you have a value of 1.8 and you have DNA, this won't tell you if it's RNA or DNA that is absorbing. So the second level of analysis should be the integrity. This slide shows the agarose electrophoresis analysis of RNA samples. What you look for are the presence of the two ribosomal RNAs, the large and the small subunit of the ribosomal RNA. And then you have to have a smear pattern in between those two ribosomal RNAs. So this would be what an intact, good quality RNA would look like. Usually the large ribosomal RNA band is more intense than the 18S. It should be about double the size or double the intensity of the 18S band. Degraded samples show maybe a little bit of the ribosomal bands and a higher representation of lower molecular weight RNAs. The capillary electrophoresis method for analysis of RNA integrity is based on instruments such as the Agilent bioanalyzer. There's another equipment now from Bio-Rad and another company that do similar analysis. And basically what they do there is analyze the intensity of the 18S, the ribosomal RNA bands, 18S and 28S. And they can provide you a relative quantitation of the sample, so you can validate your nanodrop assay values using the Agilent bioanalyzer data. And you also have an idea of the integrity. So here you also expect that the area under the curve of the large ribosomal RNA species is double that of the small band. This is what you typically get. You can get either the electrophoregram, or you can also get the gel-like image, which gives you an idea of what the samples look like. And you can see basically the same information as in the chromatogram. So this would be like what a degraded sample would look like in the Agilent bioanalyzer. And these samples all look pretty adequate. In the chromatogram, samples that look like this or different from this pattern may look like this. For example, this would be a good quality sample. This would be a partially degraded sample where you start seeing representation of lower molecular weight RNA species. And a very badly degraded sample practically shows no large ribosomal RNA subunit and a lot of RNA being in the low molecular weight. So I already mentioned the 28 to 18S. The other value that you will look for would be the 3' to 5' ratio. That would be trying to see how much of the RNA that you have has representation of both the 3' end and the 5' end of the RNAs. For that, you need to do hybridization assays. You need to be able to detect either of the two ends of the RNA. So for that, you use microarrays. You can look at the signal intensities of the 5' and the 3' ends, or you can do RT-PCR, real-time PCR, and use probes that amplify the 5' end or the 3' end of the RNA, and the ratio of the two signals should be about the same if the RNAs are not degraded. But that, I have to make a caveat here, that depends on the representation of the transcripts from a particular gene, how represented is, let's say, an exon on the 5' end and the 3' end. So you need to use, for probes, for this type of analysis, probes that are represented in the majority of transcripts from that particular gene. So I wanted to give you some data because it's kind of boring to just talk about how much RNA and all that stuff. So we've used, in my laboratory, I collaborated with Mount Sinai School of Medicine to study a recessive disorder in Puerto Ricans called the Sedley syndrome. These patients suffer from a focal facial dermal dysplasia. So it's very marked, the phenotype in the development of their face. They all have a forceps-like lesion in their temples. It's a bitemporal lesion. As you can see, the phenotypic variability is quite high in these patients. They all come from the area of Lares and San Sebastian in the island of Puerto Rico. Some of these patients have been described elsewhere, but most of the descriptions have been based mainly on what their face looks like. So together with the group in Mount Sinai, we mapped the gene to the chromosome 2, to the tip of the long arm, and we found mutations in a candidate gene in that region, the transcription factor TWIS2. In the Puerto Rican patients, nonsense mutation in codon 119 that changes the glutamine codon to a stop codon, and we also had samples from an Omani patient that also showed a nonsense mutation in codon 65. These mutations would then produce in the TWIS2 protein, which is 160 amino acids long, truncated proteins that lack the region that interacts with DNA, which is the basic region, and the helix loop helix region, which is necessary for dimerization of the protein and DNA binding. So the transcription factor we determined to be affected in these patients, we would hope then to be able to identify the target genes of the transcription factor using microarray analysis. So we use microarray, and oh, there's another piece of information. A student in my lab analyzed the protein to see, express the protein in HeLa cells to see whether the localization of the protein is affected in the mutant protein. Well, of course, we can see that only the wild type and the Puerto Rican mutant protein can be detected in the nuclear fraction, which is where it's supposed to be acting, while the Omani patient's protein is not able to translocate because it's known that these proteins require dimerization in order to localize to the nucleus. So they lack the dimerization region, so they cannot go. The protein produced by the Puerto Rican patients has the dimerization domain, so it can transport itself to the nucleus. So in terms of the microarray analysis, we use skin fibroblasts since this is a skin disorder. For the microarray analysis, we use patients from Puerto Rico compared to Puerto Rican controls, because we also did some microarray analysis of samples from the Omani patient, and we did see some ancestry, what we thought were variations due to ancestry. So out of the genes that we looked at or found differentially expressed, about 287 of them had a fold change of greater than 2 and a modified t-value greater than 6. 189 were downregulated and 98 were upregulated. We expect that since most of the literature on this protein says that this protein is a repressor, what we expected to have were more genes upregulated than downregulated. So just these numbers give us an idea that this protein in many genes acts as an activator rather than as a repressor. So, and I'm not going to bore you with details because I don't have much more time left. We validated 17 of the genes found in the microarray analysis, and they all were validated. So I'm going to skip some of this. And what I want to tell you, the last few words that I want to give you are in terms of the other side of the story. Okay, we have all this expression analysis. We also have to look at what the proteins that regulate these genes are doing. So the best type of study for expression analysis would be to look at the RNAs that are being produced, and also if the corresponding transcription factors that regulate these genes are bound or not. What is the binding pattern of the factors that regulate these genes? Or what are the chromatin modifications that are impacting gene activity? So what is known now, I'm quoting from a paper that came out in Nature Genetics in September 2009, which I found to be a very good summary. Of course, it's not available in our library, but a good friend from NIH sent me the article. The striking thing is that usually you look for regulatory elements in promoters and also the distant regions or close-by regions to the gene and to the transcriptional start site, the site where RNA synthesis starts. But from both microarray analysis looking at binding of proteins to DNA or chromatin modifications or ChIP-seq experiments where they've been doing the same type of experiment, looking for DNA binding or chromatin modifications, it is now known that Tata-driven promoters, which are the ones that I always thought were the larger amount of promoters, had to have a Tata box. They are the exception and not the rule. The other big thing that I want to give you as information, and if you can read that paper, it's much more clear there. 50% of human genes have alternate promoters. So that means there are exons that may not have been included in the microarray designs that you might not be sampling when you do the analysis with the conventional microarrays that most people are using. So there's a lot that we don't know about how genes are regulated. And when they have done these studies, they've also seen that the transcription factors are binding to sites very far from the genes and in places where they don't know what the proteins are doing there. They might be places where the proteins are stored there in the genome for later use, for modification, and then they may move to places where they can regulate gene expression. But there may be also regulatory sequences that we don't know how they work. So there's a lot that needs to be done in terms of gene regulation. And we have to look at both sides of the story, how the RNA species that are being expressed, and also the DNA binding profiles and chromatin modification profiles of the genes that we want to study. And that's where I want to stop. Thank you for your attention.